Hello students. Today we discuss about the that is our chapter going on chapter that is the ninth chapter, morphology of flowering plant. In this chapter we discuss all things that is regarding the plant, that is root, shoot, then various modification of root, shoot, then leaves. All those things we are discussing about the flowers also, and the last part, not but the least, that is a fruit. Then what is mean by fruit? Fruit in angiosperm produce a fruit. After where? After the fertilization from the ovary. Sometimes fruit is produced from ovary or without fertilization. Such type of fruit are called as parthenocarpic fruit. And phenomena is called as parthenocarpy. The example is you know very well, that is banana and grapes. Then it is C without or parthenocarp, that is a banana or grapes. Then the fruit which develops a fruit is developed as the metamorphosis or ripened ovary without or with one or more seeds. This is the definition of fruit. Then basically the fruit is developed only from ovary is true fruit, which develop only from ovary, that is a yolk. Example is mango. The fruit which develops from ovary and other flower part is false fruit. Of pseudocarp, example apple. That is, here we found a true fruit. True fruit is when a fruit is developed exclusively from the ovary of flower. Example is mango. Here, this is the mango. Then, right, we call it as a true fruit. What we call it as we call it a true fruit, because in mango we found that is a development of from the ovary. That's why we call it as a true fruit. Well, and this is the typical years of this true fruit, that is a mango fruit. We found outermost cover, that is epicard. Why we call it epicard? Epi means above. This epicard, the outermost layer called as epicard. Then middle layer is called as mesocard, and inside the inner layer is called as endocard, and inside the fruit, the final is we call it as a seed. All these things are very important when we are discussing about the true root. Then about the false fruit. What is mean by false fruit? Then this false fruit develops rather than the ovary, that is called as false fruit. Then this is example of apple. In this apple we found a pericard. We found the pericard, and the pericard outside we found the fleshy thalamus. From the thalamus itself, rather than the ovary, from the thalamus we found the fruit is developed, or the thalamus play a very important role in the false fruit. That, and the next part is, that is a true fruit has a wall, that is a pericard. Okay, this is also called as a pericard. Then, and seed pericard is further divided in outer epicarp and middle mesocarp and inner endocarp. This is also called as pericard. This is also called a pericard. Then fruit can develop from one ovary of one flower. Such fruit are simple fruit. That is, what is that? That one ovary and one flower. That is, such fruit are simple fruit. Simple fruits are further classified on the basis of their pericard. Those having thin pericard are dry fruits. Those having thin pericarp, that is they are having dry fruits. In dry fruits, the pericarp become dry and thin. Pericarp become dry and thin. In dry fruits, the pericarp become dry and thin. In it breaks open dehiscent. It breaks open dehiscent at maturity. But in some other, it does not break open. That is called as indehiscent fruit. Then, in indehiscent fruit, that is example of capsule. We are having example of capsule. That is a lady finger. That is called as a lady finger. Then, in leguminous pea, example is pea. A leguminous plant, the example is pea. Then dehiscent fruit, in fleshy fruit, that is example of tomato, has a very soft pericarp, but drop, that is a coconut, has a strong endocarp. Many ovaries of Apocarpus ganoecium can form one fruit. Such fruit as aggregate fruit. Aggregate fruit are a collection of heteros of many varieties. According to can be heteros, that is a example of strawberry also. Then heteroberries, berries, that is a custard apple. Hetero follicles. Then many ovaries of many flower, but of one inflorescence. In previous lecture, we discussed about the inflorescence. What is inflorescence? Inflorescence is a bunch of flower present on a single axis. That way, the flower they are fertilized at the same time, and because of that, we found a fruit, and that fruit is called as a composite fruit. That fruit is called as composite fruit. This fruit developed from one inflorescence. How they develop composite fruit? How they develop? They develop from the composite fruit. The one which develops from a hypopodium inflorescence is synchronous, that is fig, and psoriasis, that is a pineapple. Develops from Kotkin inflorescence. Families or group of plants having very distinguishing common characters that are we will discuss in the previous lectures. Then about the seed. First, this is about the fruit. Then we discuss about the seed. What is seed? Seed is a reproductive unit that develop from the fertilized mature ovary. Seed is a that is called a reproductive unit. Reproductive unit that develop from fertilized matured ovary. This is a seed. What is that? Seed is a reproductive unit that develop from fertilized mature ovary. Inside the ovary, we in previous lecture we discuss about the ovary. What is ovary? Here, this is the gynoecium. Inside the gynoecium, this is stigma, style, and ovary. And inside the ovary, we found the ovules. And that are the ovules, they are mature, that fertilized mature. Inside the fertile mature ovary, we found the ovules. and these ovules, they are convert into seed, and they forming a seed. Okay, then we having certain we have certain diagrams here for the endocarmic seed. Endocarmic seed, that is this kind of structure you can see here. Then this is outermost flower is called as testa. Then this kind of structures are also present. This is testa, outermost of cover. This is endosperm, and this is a cotyledons. We are having another one diagram also here. Leaf-like structure, leafy structure found in the seed, inside the seed. This is a cotyledons. Here, this is a diagrammatic diagram of endospermic dicot seed. Endospermic dicot seed, that example is castor. This is a example of endospermic dicotyledonous seed. Endosperm, and when endosperm develop from the at the time when the primary, when the at the time of fertilization, after the fertilization, one egg cell, one microspore cell is fertilized with this cell, and they form this triploid structure, and one microspore is fertilized with the egg cell, they form diploid structure. This diploid structure is developed, is converted into embryo, and this is all activity are done in ovule itself. Then here we found this 2n triploid structure is called a PEN. This is called a primary endosperm nucleus. This primary endosperm nucleus then divide and redivide to form this structure. This is endosperm. In some cases, this endosperm is utilized for the development of embryo. That is example of, we found that is a non-endospermic seed. We having the diagram of non-endospermic seed also. This non-endospermic seed having example is gram. Here the diagram. This is called as testa. Here the formation of the structures. Then we found this dicotyledonous structure having, we show here plumule and these two cotyledons, and here we found a radicle. radical here, plumule, and this is cotyledon. This is the example of this figure of, it is a non-endospermic dicot seed. Example is gram. These are the typical structure. We know it very well, and I think these are the very important to know how they look like. In your book, we don't have this kind of diagrams. That's why I draw here to understand what is meant by testa, what is meant by the endosperm, and how the endosperm is developed. This is a very important thing to know the structures. Okay. Then here this testa is a thin outermost covering. Then when we break that seed, we found cotyledons, because gram having a dicotyledonous structure, a dicotyledonous seed. Then inside the seed we are observing the embryo. This embryo having the plumule and radical. When a particular seed got a favorable condition, they are in favorable condition, they are in this kind of structure, they are developed. From the plumule there is a development of aerial part, that is stem, then branches, leaves, flower. All these kind of structure are developed from this plumule. Then from this radical, we know very well, from the radical there is a formation of the root system. There is a formation of root system. All kind of structures that is we are observed where this seed get a favorable condition. Then one question is arise in your mind, that is what is meant by non-endospermic seed and what is meant by endospermic seed. The endosperm, that is the 2n structure, 3n structure, that we call it as a 3n structure. That is structure we call it as a PN, that is called as primary endosperm nucleus. That primary endosperm nucleus, that divide and redivide to form an endosperm. In some seeds, that endosperm is utilized for the development of embryo, and certain cases that endosperm is partially utilized, and that's why we call it as a endospermic seed. And here the endosperm is utilized, that's why we call it as a non-endospermic seed. Here we found plumule, cotyledons, and radical. Then again we revise that seed. Seed is a reproductive unit that develops from fertilized matured ovule. That seed is made up of seed coat. I already told you seed coat is what is that seed coat, and one or two cotyledons, that is monocot and dicots. Outermost covering of seed is called as seed coat. Outermost covering is also called as a seed coat. Then outermost layer called as a testa. Outermost cover called as a testa also. Then the outer layer called as a testa and inner tegmen. Hilum is a scale, the seed coat through which seed attached to the fruit. Seed attached to the fruit with the help of this hilum. And that is a scar. That is a scar of that attachment with the fruit. Embryo of a seed enclosed with seed coat. Embryo is enclosed with the inside the seed coat. That embryonal axis consists of, this is the embryonal axis, consists of plumule and This embryonal axis consists of radicle and plumule. Radicle and plumule. The part of embryonal axis between cotyledons, that plumule is epicotyle, while the part between cotyledons and radicle is hypocotyle. The nutritive tissue in seed is called as endosperm. Endosperm is a nutritive tissue which is utilized for the development of embryo, but this cotyledon having certain kind of energy, that energy is utilized when at the development of this plumule and radicle, when the seed is germinate. When at the time of germination of seed, this development of embryo and radicle, they need energy, and that energy is utilized from these cotyledons, from this endosperm. So they get energy, and from that they produce a typical structure that is called as a plant. From that plant, then we discuss about the morphological structure, and all kind of structure are developed from the plant. This is all about the fruit, and this is all about the seed. Thank you.